Let's say there is some mutation that creates a beneficial allele within a population, such as lungs within fish to give them more buoyancy. And we need to get this beneficial allele out to more individuals within this population such that collectively the species as a whole can be more fit to its environment. And what this is called genetic variation. How are we going to get those good alleles circulated within the individuals of our population? And what I'm going to do in this video is walk through the 4 main sources of genetic variation. And so a lot of this is going to hinge on meiosis, the formation of gametes, of these haploid cells for humans that will fuse together to form embryos that are diploid. And so the first thing that we're going to be looking at is crossing over. And crossing over is a process that occurs during synapsis, which is this subphase within prophase 1 of meiosis. Prophase 1 is arguably the most important phase of meiosis and of this whole genetic variation process because it's when we are going to be forming tetrads. And these tetrads are when you have 2 homologous chromosomes coming together and you will form these links between them, these little bridges called chiasma. And at these chiasma, you will have alleles being shuffled between the 2 homologous chromosomes. So if you had a chromosome, the dominant allele big A, and on the other chromosome you had the little a allele, this is the point at which you could actually switch the big A and little a allele between the 2 homologous chromosomes. The next source of genetic variation is Mendel's law, specifically his law of independent assortment, which we can see is actually violated in real life sometimes when you have linked genes. But when you have genes that are very far away from each other on the same chromosome, or when you have genes that exist on entirely different chromosomes, we see this law apply. And what linked genes refers to is, what independent assortment refers to is the fact that with many genes, they don't care what the other genes are doing. And what I mean by that is if you had a gene for the height of a pea plant and the gene for whether or not the peas were wrinkled or smooth, those 2 genes for those 2 traits don't care what that other trait does because they are far enough away from each other or they're on a separate chromosome. Consequently, Mendel was able to establish a law of independent assortment. But if those genes were close enough together that during the crossing over phase they didn't separate, then we would call them linked genes. The next thing is the fact that we have something referred to as random fertilization. And what that means is if I took a petri dish and had an egg on it and then aliquoted some sperm onto the plate, 50% of the sperm right out of the gate are going the wrong way away from the egg, and by chance one of those sperm is going to actually make it first to the egg. There's going to be a fast block to entry so that you don't get polyspermy, which is when multiple sperm will fertilize the same egg, and it's a random process. You cannot tell somebody a priori which one of those sperm is going to fertilize that egg, and that introduces genetic variation to a population. And finally, what we have are mutations that occur within germ cells. And it's important that these mutations occur in germ cells because it's the only way that these things are heritable. If you get a permanent modification in a germ cell, as that cell performs rapid mitotic divisions after it's fertilized, that mutation will stick around. So if you had a good mutation for lungs, for instance, within fish, that mutation needed to have occurred within this very narrow window. If you have mutations occur within your somatic cells, that's not going to help you because that will not get inherited by the next generation of individuals. And then finally, something else I would like to talk about is Mendel's law of segregation. And what this law tells us is that gametes are haploid. What it means to be haploid is that you have one allele in that cell. You don't have a second copy on another homologous chromosome. You only have one chromosome with one allele on it. And the reason this is important is because, as we can see from this chart here, we took a diploid parent cell, and we ended up with 4 unique haploid daughter cells. If these cells were not haploid, after fertilization, you would end up with cells that could have 3 copies of all of the genes, and this is called a trisomy, which is how Down syndrome occurs. If you look at chromosome 21, people with Down syndrome will have 3 copies of chromosome 21 because during meiosis, either the sperm or the egg failed to completely pull apart the chromosomes, and this would have happened either in anaphase 1 or anaphase 2, when you had the microtubules actually pulling these things apart. And a final note that I'll make here is you'll sometimes see something referred to as genetic recombination. And genetic recombination is referring to 1, 2, and 3 above. So genetic recombination refers to either crossing over, Mendel's law of segregation, Mendel's law of independent assortment, and the fact that you'll have a random sperm fertilizing the egg. Mutations are not something that were ever intended by life. It just happens that the mutations made some kind of trait more fit, that made the organism more fit to its environment. And so I hope you guys find this useful, and thanks for watching.